Hi, everyone. I would like to welcome you. And well, thank you, first of all, for joining us and from so many different countries. I think at the moment we have people joining from almost 20 different countries across the globe. That's very exciting. So my name is Francisca, and today I'll be discussing with our guest, Dr. Eszter Nagy. She's sitting here in front of me through the virtual space. And we will be discussing the speeding up of the development of vaccines and drugs against COVID-19. So I have a PhD in biomedicine, and before I moved into the venture capital world, I've done biomedical research in different labs across Europe for more than 8 years. But even before the COVID-19 situation, I've heard from many investors that work with us at LIFELINK that they were surprised and even skeptical when it comes to this very long process that takes to develop a new drug or a new vaccine, and how long it takes to take that therapy to market, right? But scientists and founders, they wish to bring these technologies to patients as fast and as much as investors do. Nevertheless, this risk and return calculation in our heads might be a bit different from what happens to a financial market specialist, for example. So today, as an ex-scientist and here together with Dr. Eszter Nagy, I hope that with today's seminar, we can shed light on a few points. First of all, is explain you a bit how this process of developing new drugs and vaccines works and why it takes so long. And then discuss a bit how we can speed up this process and how scientists try to mitigate risks and accelerate it. So I will start by giving you a brief introduction on the science and about the virus, just to tell you some concepts that will help you understand better today's discussion. Then I'll invite Eszter to answer some of the questions that we think it's important to clarify. But at the end of the session, we will be going through your own questions. So please feel free to write your questions in the chat or in the Q&A box, and we will be going through those at the end. So with that, I will start by giving you that introduction. I think we all know by now that SARS-CoV-2 is actually a new virus. We didn't know it before this crisis. And we know that it spreads to droplets, for example, when we cough or when we talk and we're in close proximity with others. So once we've been in contact with the virus goes through our body and it starts to try and infect our cells. Now, SARS-CoV-2 is covered by a protein that is called the spike protein. And this protein, what it does is bind to a receptor in human cells. I think a lot of you have heard about the ACE2 protein. So this protein is very present in lung cells and also in heart cells. So the spike protein is able to bind this protein in our cells, and it's through this protein that it actually enters our cells. Now, a virus itself is not able to replicate on its own. It doesn't have the machinery and the tools inside the virus to actually multiply. So what it does, it takes advantage of other cells, like our cells, to multiply. So in this case, SARS-CoV-2 enters our cells and releases this information, this viral RNA, that encodes for proteins, for viral proteins. What it does is that it takes advantage of our own tools, of our own cells, and start producing these viral proteins. Through this process, it's actually able to create new viruses within our cells that are then released and keep on spreading throughout our bodies. Now, we know that in severe cases of COVID-19, there's a lot of mechanisms, a lot of molecular things happening. We know that, for example, our immune system is, of course, trying to fight the infection. We know that we have immune cells producing these immune factors that we call cytokines. They are supposed to be fighting the inflammation. However, when we have a very exaggerated inflammatory response, we can start to damage our own tissues. And in this case, we see that in severe cases of COVID-19, we can have a very strong production of cytokines that leads to a deregulated immune response. And we call this a cytokine storm. It's very high levels of cytokines that instead of helping fight the virus, they start damaging our own tissues. So we also know that another thing that happens in severe cases of COVID-19 is that certain specialized immune cells start to lower in numbers in our bloodstream. So altogether, this ends up contributing to us not being able to fight effectively the virus. And as a consequence, we observe in these cases lung damage, acute respiratory distress syndrome, this being the leading cause of mortality in COVID-19. We see systemic inflammatory response syndrome and cardiac failure. And all this together leads to a very low survival rate. Now, what we're all trying to find out is how we can actually stop the virus. So we know that at the moment there's no vaccine, no antiviral specifically developed for SARS-CoV-2 that has been approved yet. We know that if we want to prevent this virus to infect us, we need to develop a vaccine. A vaccine is going to educate our immune system and prepare it before we come into contact with the virus. On the other hand, if we want to treat patients that have already contacted with the virus, we can use antivirals that are going to block the activity of the virus that has injured our cells, or we can use drugs that are going to treat the disease. So, for example, attenuate the consequences of this infection. So when we look at the vaccines, there are many different approaches that we can take. We can develop many different vaccines, but the most common approach would be to take the information that we have inside this virus. We can take a little bit of it that encodes for this spike protein that I told you about, and we can use it as a vaccine. So we can train our immune system, we educate it, we show the spike protein that alone is is harmless, and our immune cells learn and get acquainted with this spike protein and start producing specific antibodies against it. And what this means is that even before we've seen the virus, we already have antibodies against it. So that when we do encounter the virus and we do have an infection, our immune system is completely prepared and is able to fight off the virus. So this type of vaccines would be the best approach if we want to prevent new cases of the virus, of COVID-19. On the case of antivirals, as I told you, they block the activity of the viruses. And at the moment, there's a lot of labs and a lot of institutions developing different approaches of antivirals. We have, for example, people trying to stop the binding of the virus to this ACE2 protein. And on the other hand, we have people that are trying to block this replication complex. So they're just trying to stop different steps of this process of replication of the virus within our cells. In the case of treatment of disease, we can try and, for example, attenuate this exacerbated inflammation, as I told you. So a very common approach is to try and block or inhibit the cytokine called IL-6. So we know it plays a very important role in this cytokine storm, and there are a lot of people trying to find a way to calm down this very exacerbated inflammation in the body. So as I'm telling you that there are a lot of different approaches and a lot of people trying to develop new therapies, it's important to understand how these drugs and vaccines are actually developed and to understand why it takes so long. So here I'm showing you the normal process of development. So I'm not taking into account a crisis like the one we're facing at the moment. But it's just for you to see that in normal circumstances, this process takes a long time. So first of all, we need to start by identifying our target. So for example, we can take IL-6, the cytokine I just told you about, as an example. You can decide you want to target IL-6, you want to block it. So first you need to develop a compound that actually blocks it. And this is done in the lab, and it can take many years of research until you finally get your compound that is efficient enough. And first you're going to test it in cells. You're going to show in cells that it actually works and it does block your target. And then you're going to move on to animal studies. So before you even reach the clinic, you need to show that your compound actually works and at least seems safe in animals. And this process can take from 3 to 10 years of research in the lab. once you get to the clinic, the first thing you do is you need to show that your compound, your drug, is safe enough. So here what you do is you take very few patients, you start by testing in a very low amount of patients, and usually these are healthy volunteers. These are people that don't have any conditions that are going to put them at risk in case your compound does have side effects. So you test for safety, and here it can take from 1 to 2 years. You then move to a phase 2 of clinical trials. Now you can take a higher number of patients, and here you take people that actually develop the illness you want to treat. So you want to show that your compound is safe, as you've seen in phase 1, but it's actually efficacious and is better than what's out there in the market. You're going to take about 100 to 300 patients that are sick, and you're going to demonstrate how much better they become when they're treated with your compound. Again, this can take between 1 and 2 years. And then finally, you move to phase 3. Phase 3 is a large clinical study. You take many, many patients. You take different kinds of patients as well, because you need to be able to show you need to be able to detect if there are any side effects across different types of patients. And here you're going to confirm again efficacy and safety. And because this clinical study is the last one, is a very big one, depending on the time it takes to recruit all these patients, it can take a long time. So we're talking about between 2 to 5 years. And only then, with all these studies and scientists and regulatory agencies being sure that your compound is safe and it works, only then can it be approved and start to be commercialized. The only difference, or the main difference between a vaccine and a drug development, is that in the case of the vaccine, you only take healthy volunteers throughout the process. And this is very important because, as we will discuss with Eszter, it means that you need to take safety even more carefully, because you don't want to put at risk people that are completely healthy. Now, the other important thing that you should know is that regulatory agencies play a very important role throughout this process, and they're the ones that decide whether your drug or your new therapy goes forward or not. So here I marked all the steps where these agencies are going to evaluate your data and decide whether you can move on with this new therapy. So as you can see, there are many different steps where they're going to look and they're going to check whether your data is convincing and it's solid enough. And with this, I would like to introduce you our today's guest. I would like to tell you a bit more about Eszter. So Eszter is the founder of CEBINA. CEBINA is a Central European biotech incubator and accelerator. So Eszter has over 9 years of academic research, more than 20 years of biotech experience. She has published over 80 scientific articles and books and has contributed to about 40 patent families. She has a strong background in anti-infective immunology, prophylactic and therapeutic vaccines, immune adjuvants, and anti-infective antibodies. So prior to founding CEBINA, she was the SVP of research at Intercell, where she was responsible for 20 infectious diseases and vaccine development projects. She was also the co-founder and CEO at Arsanis, a company that IPO'd at NASDAQ, and where she developed anti-infective monoclonal antibodies. And in the meantime, she has also co-founded several other companies. For example, the vaccine company Evelicure Biotechnologies, and other companies such as Calixa Biotechnologies and Algonist Biotechnologies. And at the moment, as I told you, she's the founder of CEBINA. CEBINA is a Vienna-based biotech incubator and accelerator. And the companies that go into CEBINA basically have access not only to lab space, to offices, but also to a lot of know-how and services that help them develop and mature. And so they have a mixed model between incubator and accelerator that helps companies and new formed startups gain momentum, accelerate this maturation process, and actually increase the survival rate of startups that we know that is usually very low. Well, and with this, Eszter, I think we can start. So today we have some questions we thought would be important to mention. I think as I was saying, there are currently no treatments against SARS-CoV-2. However, we see a lot of resources being developed to instead of being used to develop new drugs. We see a lot of pharma companies screening through their portfolio of existing drugs and trying to understand if they can repurpose some of those drugs for the treatment of COVID-19. I think it would be very interesting if you could tell us a bit, why are these companies trying to repurpose drugs rather than create new ones that are specifically developed for this new virus?
I think they try both, but the answer to your question is very simple and short: time. So we need a cure now. COVID-19 is a new disease, obviously we don't have treatment for that. As you explained, the product development process is very complex and takes years. We don't have years to develop new drugs. So therefore, the pharma industry, but I would add that also the scientific community is focusing on drug repurposing. What drug repurposing means that you look at drugs which are already approved, marketed, available, and you ask the question whether there are drugs which could be effective against COVID-19 as well. Actually, this approach has been proven to be feasible. In the last couple of years, several new medicines have been introduced through drug repurposing. Obviously, the pharma companies have an incentive to do that. Once the products are not protected by patents anymore after 25 years, they try to find new medical use, so extend the patent life. That's one driver. But in this situation, I think the driver is really time. So it may sound counterintuitive that we look at drugs which were developed for another disease. What can be the rationale that you find drugs against COVID among those? I think there are several approaches how you can find such drugs. The most obvious one is based on the known effects of drugs and the biology of COVID-19, and you try to match the activity of the drug with the disease mechanism. So the most obvious ones are the antivirals, because viruses share a lot of features. Why they are, for example, when they are replicating in our cells, they use quite simple machinery. And if the virus, which is efficiently tackled by a drug, is sufficiently similar to COVID-19, I think there's a good chance that that antiviral drug would also work against COVID. The best example is remdesivir, which is also very frequent in the media, which was developed against Ebola, and now it's being tested in a phase 3 trial against COVID-19. The other class of drugs are the anti-inflammatory drugs in the case of COVID, because we know that the most severe side effects or a more severe form of COVID-19 is the so-called cytokine storm. As you explained, when our immune system overreacts to the virus, and instead of being helpful, it's harmful to the tissues, especially to the lung. So obviously, then you look around among anti-inflammatory drugs, and then you ask the question whether those can be helpful for COVID-19 as well. The other approach is more scientific, I would say. Network biologists and experts who developed in silico prediction methods now apply their platform to try to match the cellular pathways which are activated by the virus with those which are also affected by the drugs. And I think that's a very interesting new pool of drugs.
Yeah, this would be basically using computer power and programming capacities to...
Big data, using experimental data, and I think that's the other very positive development in the last couple of days, that scientists publish their data very quickly. Manuscripts are online before they are peer-reviewed. So basically the scientific community changed its attitude immediately as a response to the crisis. and they want to help the global community by sharing their knowledge. And I think that's the other source of repurposed drugs.
Yeah, and you mentioned already one of the very, I think a lot of people hear about many repurposed drugs, but I think another one that everyone has heard of is also chloroquine and hydrochloquine. However, I think a lot of people are asking themselves, if they heard such good news about the use of this drug, why isn't that it's not widely approved through different countries? So I think it's important to understand, why is it that the regulatory agencies are not yet allowing for people to be treated with, for example, these anti-malarian drugs in every hospital across the globe?
Yeah, it's quite a big controversy. And I think the main reason why these drugs are not approved yet is the uncertainty whether they are effective and whether there's more benefit than harm using them. Obviously, there is a reason why chloroquine and hydroxychloroquine are considered as anti-COVID drugs. So these compounds were shown to have antiviral effect in vitro. So basically, it slows down the viral propagation when you test it in a test tube. And it also has an anti-inflammatory effect. And that's the reason why hydroxychloroquine is used in autoimmune patients, in rheumatoid arthritis and lupus patients. So there is experimental empirical evidence that with dual mode of action, these drugs could be beneficial. However, there's no definite proof that actually it really saves life. There are several small clinical studies where benefit was shown, especially in the lung inflammation, while in other studies they could not demonstrate any benefit. I think the most concerning is the side effect profile of hydrochloroquine and hydroxychloroquine, because besides gastrointestinal and skin problems or side effects, it can also cause permanent reduced eyesight or vision. Obviously, it's not so concerning if you really save lives, because it's better to see less but still alive. But there's also potentially fatal side effect, and that's the irregular heartbeat. And actually, that's the reason why the most recent studies in Brazil were halted, because in the higher dose of hydroxychloroquine, several deaths occurred. So immediately the study was stopped, and unfortunately the lower dose group could not be evaluated because it wasn't sufficiently big enough to give statistical power for the data. But I think there are two camps: people who really believe that it's working, and the others who are skeptical, and they are really waiting for data. But by the way, FDA approved the use of hydrochloroquine and hydroxychloroquine in case a clinical study cannot be performed. I think the other issue is the capacity of clinicians to run these studies. For example, in Italy, doctors are really struggling taking care of the severely sick patients, so it's difficult to perform well-designed trials with sufficient large number of patients. So there are several hindrances. I would say that we are a couple of weeks from having more conclusive data on chloroquine, hydroxychloroquine, because there are several studies running in several countries. And now there's an international coalition in this fight, and the studies are more or less aligned. So it would be possible to combine data from different countries, and hopefully we can conclude.
Exactly. And soon we will know the dose and also what stage of the disease will be better to use this drug.
Exactly.
Right? Because I think that's also part of the risk-return assessment, right? You need to understand how much do you want to risk. It also depends how advanced the disease is in a patient, right? So if the patient is quite sick, then you might be able to risk a bit more and try a higher dose.
Absolutely, that's right. I think we also have to understand physicians whose primary guide is primum nocere, first of all, no harm. So obviously, if there's a suspicion that you can cause more harm, for example, you can cause the death of a patient because of an irregular heartbeat, then you're hesitant to routinely administer the chloroquine at high dose to counteract the virus. So it's always a balance between the risk and the benefit. I think it's especially true for new drugs and vaccines. As you mentioned, vaccines are given to healthy people, so their safety is even more concerning than in the case of a potentially life-saving drug. So I'm sure there will be lots of compromises, and as soon as the efficacy is shown, then playing with the dose will allow physicians to use chloroquine. I would, however, warn the general public taking the medicine, just buying in the pharmacy what you can because you don't need prescription, and just taking it as a preventive measure. I don't think it's the right approach because the potential side effects are severe and individuals may react differently to these drugs.
Okay.
I think it should be used in hospitals where the treated patients are under surveillance.
And hopefully we'll know more also in the next few weeks. So we will know better, what the doctors will know better as well, what dose should be used and what type of patient. Yes. And so when we talk about repurposed drugs, how long do you think we will need to wait until we see these reach the market at the global level?
Yeah, I think in the case of chloroquine, hydroxychloroquine, I think it's a couple of weeks. For other drugs, couple of months, I think. In the case of repurposed drugs which are in development, probably 3 to 6 months. Example what you mentioned, the IL-6 antibodies, anti-IL-6 approaches, which were already on the market or in late stage clinical development, or remdesivir. So here, the time till completing the efficacy trials and approval is expected to be, let's say, 3 to 6 months.
Okay.
It's still much, much shorter than the development timelines for new drugs. So it's still, I think, it would be unprecedented if the scientific and the medical community could come up with these new drugs in that time frame.
Yeah. So actually following up on that comment, when we think about new drugs that are being developed, as you said, the process is going to take a lot longer to develop these specific antivirals. But can we cut any corners? Can we make the process that I just showed a little bit shorter somehow? What can be done to make these get closer to the market in a shorter period of time?
I think you can certainly chip off time at in every steps, but there are limitations. Obviously, here you show this slide again. I think it depends where the drug joins the development path. Let's say there are already in vivo studies, then the phase 1 has to be done and efficacy has to be shown. In the case of new drugs for COVID, I think what can be really reduced is the efficacy trial time, because now I think these development projects are prioritized in every testing site. So other drug developments can just follow this. And how you can shorten this if you allow smaller studies. So for example, I cannot imagine that a phase 3 trial with several thousands of patients would be required here by regulatory agencies. So smaller trials means shorter time. Obviously, it also comes with a smaller safety data package. So again, it goes back to the risk-benefit, but for patients, for a curative drug, for a life-saving drug, I think this is completely acceptable. If it's a vaccine, I doubt that you can develop a vaccine with smaller than 1,000 patients, for example, in phase 3. And I think that would be probably the absolute minimum. So it depends on the type of the drug. And regulatory agencies, I'm sure they will respond quickly. That's the other aspect which is not considered to reduce the time till approval.
They will likely prioritize this type of studies and
Absolutely.
be quicker to review it and decide on approval.
Absolutely. I think the other important factor is manufacturing. So obviously, if you repurpose an orphan drug, which was aimed at a very small market, and the production scale is matching this market, so it's small, it takes time before you can scale up the manufacture to basically supply the world population with a preventive or curative drug. So that is also a considerable factor.
Mm-hmm. And apart from hydrochloquine, what other drugs should we keep an eye on?
Mm-hmm.
Could you tell us a bit about? We've heard about a lot of them in the news, but what are the ones that for now have shown a bit more promising results?
Yeah, there are many, and I think you may want to show the slide which summarizes actually how quickly and robustly the pharma industry responded to this crisis. So there are several hundreds of programs, at least reported, and many of them are in clinical testing already. We mentioned antivirals as one of the obvious drugs for repurposing or to be developed. And here I would mention remdesivir, which was developed against Ebola. Ebola is also a enveloped RNA virus, so I think the similarity is sufficient enough to allow this to work against COVID, and obviously there are already experimental evidences that the efficacy trial will be likely successful. We have to see what is the level of efficacy. But in that case, I think the efficacy requirement will be lower. So even if it's not 80-90% curative, but only 50%, that would be still accepted. The other big group of new products are antibodies.
Mm-hmm.
And here, I think antibodies, we have to differentiate between two main categories. Antibodies which are targeting the virus, either monoclonal antibodies or the plasma transfer, which is also very often in the news and which is likely to work. And in a short time frame, I think that has a much higher chance to reach the patients than new monoclonal antibodies. So these are the antiviral antibodies which are given as a passive immunization. So basically, you are provided with an effective immune defense against the virus. The other category of antibodies which are meant here are targeting our own immune system and aims to tame this overshooting inflammation which is caused by the virus. For example, the anti-IL-6 approaches. So it's a mixed group, but I think both are very promising.
And for example, the plasma transfer approach that we've heard of, what do we know of it? Could it be also a promising approach?
I think that it's also quite imminent. The problem with the plasma transfer or hyper immunoglobulin transfer that you have to take it from people who already overcome the COVID infection.
Mm-hmm.
For example, in China, there are 70,000 of these people, but there are individual responses to the virus. So we don't know whether everybody was able to mount an effective and protective immune response. The antibody levels will be different. So the batch-to-batch variation will be different. And that's why it will be, I think, a bit complex to really evaluate it, but it's likely that it will work.
It could be an approach.
But it has limitations because of the number of people who were involved, and hopefully we won't have other resurgence with this similar spread, which means that your supply, your blood supply is also smaller, because the antibodies will also wane with time, which are present in the serum.
And another question that we've come across is, if some of, for example, the antiviral drugs that are being tested at the moment, if we see that they work better at earlier stages of the disease, COVID-19, do you believe that at some point it will be possible for doctors to prescribe the drug to the patients that are going through these mild symptoms at home?
You mean the repurposed antivirals?
Yes, exactly.
I think it depends on the known side effects of such drugs. If they are severe, I'm sure that these can be given only in hospital setting, or if the repurposed drug has not a side effect, but the main effect, which becomes a side effect in a healthy person. For example, hypertensive drugs are also suggested. Losartan, for example, is being tested. It's an anti-hypertensive drug. So if you start giving it to people who have normal or low blood pressure, obviously, then it will have a major side effect. So I think it's not that likely that the drugs which were developed for another disease would be given to people who are outside of the hospital. But I think it completely depends on the type of drug.
And for example, if we focus on vaccines for a moment, so we know there's a very high number of vaccines projects that have been reported and that seem to be being developed at the moment. Do we know more or less when we can expect to have a vaccine globally available?
Yeah, I think we hope, and everybody hopes that it's weeks and months. I think it's impossible. I think it's just a dream, what we've seen in the movie Contagion, where basically within days or weeks the vaccine was available. My guess would be a year and a half. I think that's the earliest when we can have a vaccine which can be broadly distributed. Even the ones which offer a very fast development path, for example, the mRNA-based vaccine from Moderna, which is a new technology, and it has been developed and supported by public and NGO funding, exactly because of its the advantages it offers. Basically, you inject genetic material mimicking the viral mRNA, the viral genome, into people, and the vaccine antigen, for example, the S protein, will be expressed in our body. And then our immune cells recognize this antigen and then produce antibodies. So basically, you don't have to produce the protein in a factory, because your own body will do that. But still, this technology, although I think it's very promising based on animal data and initial human data, there's no experience with such vaccine. So we have to see. But there are a couple of other vaccine candidates which are approaching the clinic or has started clinical development. I think at least a year. For example, Moderna announced that they started the vaccine study in March, and they expect the final study results in June 2021. And if that's one of the fastest, then I think you can extrapolate, and then probably I can defend my time frame that I gave here.
Exactly. So But then, if we're expecting these vaccines, the first ones to get to market in a year and a half, what role can we expect these vaccines to have, given the time frame and also the crisis that we have at the moment?
Yes. But I think it also gives a good justification why we focus on drug repurposing, what we just discussed.
Yes.
And that the real handle of this pandemic is expected only after a vaccine is introduced. It's because the current measures, quarantine, and also the travel restrictions, the social distancing, achieve a decrease, significant decrease in COVID-19 infections. But as we go back to normal or semi-normal, it's expected the virus will come back. So we will experience the different peaks until an effective vaccine is available. Unfortunately, it's unlikely that this virus will show seasonality as a normal coronavirus, which causes common cold or the seasonal influenza, as it was also seen with the 1918 Spanish flu. It peaked in the summer months, which is quite unusual for the flu virus. But here, we are not dealing only with the survival of the virus in the environment, but with the infectivity of people who are carrying it, either with symptoms or without symptoms. So until the virus is in the community and we increase the social contact and we allow the virus to spread, it will. So I think the other concern with the vaccine, that it will be specific for this virus only.
Yeah.
And it won't really address potential other future pandemics caused by coronaviruses. And as we've seen with the SARS and MERS in the last 15, 17 years, now it's the third time that the coronavirus occurs as a pandemic. So I think we can extrapolate that it may happen in the future as well. So the question is whether with this vaccine we are doing something for next pandemic preparedness. I think it depends on the type of virus which comes back. If it's sufficiently similar to this SARS-CoV-2, then we would already have an effective vaccine. For example, if the SARS vaccine in 2000, which caused the pandemic in 2002, if we had a vaccine against that virus, because most of the virus programs were stopped as the virus disappeared, there was no economic impulse to further develop them. That vaccine would be at least partially effective here, because the SARS and the SARS-CoV-2 is sufficiently similar. But for example, a MERS vaccine would not be effective, because the S protein and other antigens and other proteins in the virus are too different. So it's not, it's very unlikely.
But the fact is, the development of this vaccine was actually stopped, because it meant it wasn't worth it developing them anymore, right? Economically.
For the developers, but as part of the preparedness, you can develop the vaccines. You can take it through the whole development process. You can have it approved. So if, when the next pandemic comes and that virus shares sufficient homology and similarity, then simply you just scale up the process, but you already have an approved process and an approved product.
I guess what you're saying is that the best would be to have a vaccine that would help us prepare for future pandemics and would not only help with this one right now, but also with future ones.
Yeah, so the dream vaccine would be a pan-corona vaccine, which would be able to induce antibodies against all coronaviruses, which can resurface or come out from reservoirs where they are now. Yes. Most likely in bats and pangolins and other mammals, which can serve as an intermediate host. And I think there are, I see significant efforts towards that direction, meaning that several thousands of coronavirus genomes are deposited, not only from humans, not only from this current pandemic, but also from other sources. So that would be one way how you can rationally design a complex antigen which represents several coronaviruses. And then I think that would be a vaccine which would really contribute to world preparedness for the corona pandemic in the future.
Okay, no, I think that's definitely an exciting approach, and yeah, it would make a huge difference then for future pandemics. And I think another very important lesson that I think we've learned from this crisis is perhaps how unprepared health systems and societies were for such a pandemic. And as a result, we've seen that different countries have taken very different approaches. One of them, for example, I think has raised a lot of polemics. For example, when we talk about herd immunity, I think it would be great for you to also tell us a bit more how that could potentially be a good or a risky approach at the same time.
I think herd immunity would be effective, that's for sure. I think herd immunity means population immunity, and basically it provides indirect protection for those who are not immune to a certain pathogen. But to achieve that, and there are always individuals like the newborns who have not been vaccinated or not exposed to the infection, or the vulnerable populations, immune compromised, or those who refuse to immunize their children, for example, against childhood diseases. So I think herd immunity would work, but it has a price. You sacrifice those who are vulnerable now. And obviously it's an ethical, moral issue whether you can do that. And I think the other question is what level of herd immunity you need against COVID-19. I think it depends on the so-called R number, the reproduction number. I think everybody heard a lot about this. It's estimated for for the SARS-CoV-2 virus is estimated to be between 1.5 and 4. So it's a quite broad range. But if we take an average, a median, let's say 2, then you would need 50% herd immunity, meaning that 50% of the society, of the population, is immune against the virus. Then those who are not immune would have significant protection. If this R number would be, let's say, 10, if it was a very aggressive, very permissive virus, then you would need 90% herd immunity to really stop the infection endemic or pandemic. So I think it would work, but again, it has a price.
Yeah.
And for example, in Sweden, they argue that their social behavior is different from other countries. For example, from Italy, where several generations live together. So the most vulnerable, the elderly in Sweden, usually don't live in the same household with their children or grandchildren. So the social distancing, what they are practicing on a daily basis would protect them from a huge disease burden. I think time will tell us whether it's true or not, but in most countries, I think the conclusion was that you cannot allow the virus to cause herd immunity. You have to slow it down. You have to match it to your healthcare system's capacity, and you have to do it until a vaccine is available. So instead of the virus, the vaccine would induce the herd immunity.
I see. No, very interesting. I think we still have 10 minutes for questions from the audience. I've been taking note of some of the questions that were sent to us. I think one relevant question has been, in practical terms, how is it going to look, how the real life is going to look after, well, in the next few weeks or months, and how do we expect to go back to our normal lives, how slow that process might be. Could you share also your perspective on that, or how do you expect things evolving a bit from now on?
So I expect that as soon as the number of new cases decline and we control the virus, there will be normalization. So certain restrictions will be lifted. But as soon as we experience the resurgence of the virus, those restrictions will be implemented again. I think the most severe restrictions for a longer period of time will be on travel. So countries who have managed or managing to control the virus in their population cannot afford to allow people bringing it from other regions, especially when it's less controlled. So I think that will be so international travel will be significantly reduced in the next 6 months.
So probably we will need to wait for a vaccine
until autumn or so. And also we have to see how the virus behaves during the summer months, whether it shows seasonality or not. This virus is an enveloped virus, meaning it has a lipid bilayer and it's sensitive to heat. So it survives for a shorter period of time. But again, if people are carrying the virus, it doesn't matter whether it's 2 days or 3 days, because the source of infection would be the human individuals.
So it might take some time. For example, once we have a vaccine, then we should slowly be able to go back to our normal life. But until then, until we don't have a way to stop the virus from spreading, it will be very difficult, right, to go back to a very normal life as we knew it.
I think so, at least based on the current knowledge of the disease and the virus. But we have to see in the next couple of weeks and months, and how what is the situation after countries start to lift the restrictions, and how the virus responds to this.
Yeah. We had another question about the most promising drugs or vaccines against COVID-19, but I think we covered that one. And then another question we got is about the testing procedures for COVID-19. So I think a lot of people are wondering, why is it not possible yet to get a quick way and cost-effective way to test the patients and diagnose them, basically?
Again, I think it's time. So I think the manufacturers are responding to this demand for sure. And there are tests which are easier to be performed and quicker than the first ones. And also the precision of the tests are improving. So there are some antibody-based tests which were which showed false positivity, because probably they were not specific for this virus. I think the PCR-based, the genetic-based tests are the most accurate, but obviously they need a device to read the data. But there are, I think, companies who are responding with portable devices to this PCR-based test. And I think we will see soon that shortages won't be the explanations why certain countries do not test.
And what happens, for example, when we want to know if a person, for example, asymptomatic or that showed only mild symptoms, can we show later on, can we test for that person and know if they had indeed COVID-19 and they were actually recovered or developed immunity? Is there a way to test for that?
Actually, there are the first reports suggesting that those who already suffered from the disease in different forms can carry the virus asymptomatically. It's something worrisome, because they are then reservoirs of the virus. And then the question comes, what is really the protective immunity against the virus? So I think we still need to have more information from those studies to really understand whether those individuals had sufficient level of protective antibodies, and what is the reason that the virus may survive better. The virus responded by changing its antigenic structure, which has not been reported yet. So this virus is not so unstable. It hasn't changed its infectivity or antigenic structure, at least based on the global tracking. But certainly that's something what we have to watch in the next couple of weeks and months, because it can also educate us about the type of vaccines we need to develop.
Mm-hmm. Another question that I've received here is also why is it that, for example, chloroquine is being taken against malaria with no special precautions, and now it's being so complicated to use against COVID? I think people are quite confused, since this seems to be a drug that has been approved many years ago and has been used by many people. Can you tell us a bit more about that as well?
Yeah, I think it goes back to the risk and benefits. So for a long time, that was the only anti-malaria drug. So obviously, malaria is a very concerning infectious disease. So the not too favorable side effect profile of chloroquine was accepted against malaria. I'm not saying that the COVID-19 would be different here, but since this drug was not specifically developed for COVID-19, it seems that potentially higher doses of chloroquine are needed to be really effective. At least in the clinic, several higher doses were tested. And above a certain dose, I think there are now proven safety concerns. So those high doses shouldn't be taken. Plus, there are also certain individuals, a vulnerable fraction of the population, who respond to even to the normal dose of these drugs with adverse reaction. So I think that's what I would add to this discussion. But it's a good argument. And again, if you are severely sick and there is efficacy shown, I think it should be given.
Yeah, I think
but not as a preventive measure for the general population.
Yeah, exactly. And actually, I think chloroquine has actually been replaced by other anti-malaria drugs, exactly because of the side effects it has, right? So people have actually been trying to replace it.
Yes.
Of its side effects. So I think, as you said, it's again that risk and return assessment, because if it's only effective at very high doses, but also all these side effects are there, then you really need to select very carefully the patients that take it. And we need to be very sure that it's very helpful, because otherwise, why would you take it?
Absolutely.
Yeah. I think we also got another question more related to the mortality rate. It's more of a comment as well. It says that the Spectator has published an interesting article last week suggesting that there might be some error in the presumed mortality rate of COVID-19. According to them, not only most countries are reporting death using different standards, but also most of the testing is done on a sample population made directly in the hospitals, where some of the patients are already worse in condition versus the rest of the population who are not being tested. And so what's your view on this? I think this is a very important one.
It's absolutely true. I think there are certain countries who are really proud that they are world leaders in the low mortality rate due to COVID-19. For example, one neighboring country reported 2 deaths among 800. So it's quite unbelievable. So I think it depends what you report on the death certificate and whether you really test. But I agree, and I think it's controversial. I'm sure that the capacity of the intensive care beds and the respirators are crucial in reducing the mortality rate. Access to healthcare, absolutely. The age tree of the population. For example, in Italy, 20% of the population is over 65. As I mentioned, they live together with the family members who had more chance to contract the disease, and probably that's one reason why the mortality rate in Italy, besides the not inappropriate capacity of the hospitals, contributed to the high death rate. But it also depends on just reporting. In general, I think we consider that 15% among the elderly, COVID-19 leads to death. why below 1% between 40 and 50. So it's really a question of age, and I think that's the other very interesting aspect of the disease. And it would be very important to understand why children and young people are kind of resistant to the virus. They get infected, they can carry the virus, but they don't develop the disease, or most of them. Obviously, there are sporadic cases, but in general, that's true. And if you understand what is different in the young tissue versus in the old tissue, what the virus is sensing, could be also another way how we deal with this type of viruses.
Yeah. Well, yeah. So unfortunately, we're running out of time. I think it would be just interesting, as you were saying, because one of the questions was related to the amount of fake news and rumors circulating in the media. I think it would be good to give an overview of some of the questions that we still don't have an answer to. So it is a new virus. There are things that we still don't know. And I think it's important that people know that. So when they read news, they understand, okay, it's not yet clear if this is true or not. You just give an example. So why is it that younger people don't seem to show symptoms? Could you tell us about other things that we still don't know about this virus?
I think there are many things we don't know, and telling the truth, the breadth of protective immunity, whether the S protein, for example, would be sufficient. I think 95% of the vaccine approaches now are based on S protein. We don't know whether pre-existing antibody is potentially enhancing or protective. Coronaviruses are quite common. And then they are seasonal, so they are a common cause of common cold. So one can imagine that there are certain cross-reactivity among certain strains, and this SARS-CoV-2 virus. So I think we need to analyze the antibody repertoires of those individuals who are less or more susceptible than the general population, and make some conclusions.
And how about reinfection? Do we know if once we've come into contact with the virus, developed immunity, do we know already if it's possible to be reinfected or not?
I think that's what this recent small study from China would suggest, that they identify the virus also in those who already went through the disease.
So that would highlight
that will be very important to understand.
So that will, if that's the case, if we can get reinfected, it would be even more important to develop a vaccine, because that means that after fighting the virus, we can still, again, be infected and face again the disease. Well, Eszter, wonderful. I think we've not been able to cover all the questions, but unfortunately, we're running out of time. What I ask everyone that has more questions or that we haven't been able to cover them, please just email us. We'll be going through all the questions. And you have our contact as well, I think, in the emails you received about the webinar. So please feel free to reach out. And with that, I would just like to give a big thanks to Eszter for joining us today. I think this has been fantastic. And this discussion, I think, has cleared a lot of wrong ideas out there. So thank you very much, Eszter, for today's participation.
Thank you, Francisca.